Well, hello everyone. Good evening and welcome to Cybernetics Live. I'm Peter Tuddenham. I'm the current Vice President of the Cybernetic Society. It's my pleasure to get you all started here tonight. A few words about the Society. The Cybernetic Society was founded in London in 1968, and in 1976 it was registered as a specially authorized society under the Friendly Societies Act of 1974. So it's been a friendly society for some 50 years now, and that word friendly matters. The Cybernetics Live session is the culmination of a whole series of events, and for me it's an example of what a friendly society is really all about: respecting and honoring one another and supporting each other in whatever way makes sense. So what I would like to see is more of our members developing their own work and then sharing it with us in future Cybernetic Live sessions, exactly as Sally is doing tonight. And if you're watching tonight or you're on the YouTube recording and you're not yet a member, please do join us. It's only £30 a year and you'd be very welcome. That's a spirit I hope carries forward tonight. So I will moderate questions and answers at the conclusion of tonight's presentation. Please use the hand raise feature on Zoom. This session is being recorded and will be on YouTube, on the Cybernetics Society YouTube channel. If you do not wish to be on the recording, please turn off your camera and microphone. So we'll go for 90 minutes, and then there will be a later in the bar period after this first 90 minutes when the recording will be stopped. So now I'd like to hand it over to Richard Berry, who is a council member, to talk a little bit more about how we got here and to introduce Sally. Richard.
Thanks very much, Peter. I'll keep it brief. Remembering back in September, Vlatko Vedril gave an inspiring masterclass in relation to his specialism on quantum information theory. And in this 100th year of Stafford Beer's existence, he would have been a centenarian, I believe, this year. He coined the phrase later in the bar, and a group of us met later in the bar to discuss Vlatko's input. What was really interesting for me is the way the group developed, the dynamics, the conversations, and the extension of knowledge. Along came Sally, and just kind of blew us away, really, with her dramatic input. And we're talking about the interface of some real complexity, quantum mechanics, chemistry, physics, biology. They're challenging enough. But then we add some more variety and we add cybernetics to the mix. The insights are provoking. The possibilities are profound. And to cap it all, and I think this is the lovely piece, is Sally was actually supervised by Stafford in her initial doctoral research. So really looking forward to it. Welcome, Sally. And I also know there are a number of people who can't make it tonight, but are going to catch up with the session later online. So you're in good hands, and look forward to hearing it. Thank you.
Okay, thank you very much, Richard, for the introduction. Whilst I'll leave Brian Josephson's book title on the screen while I do just a couple of bits of housekeeping. One of them is that I've been told that my presentations are quite intense, a bit like being machine gunned. And so in the spirit of the old program, Allo, Allo, I would say advise you for that to listen carefully. I can say this only once. The other thing is a paradoxical thing you need to sort out for yourself is, are you sure you want to see this? In the sense that once you've seen it, you can't unsee it, and it will peck your brain. So we'll go to the presentation. Vlatko made clear the need and was wondering if cybernetics can help to find a fresh perspective by which they might approach quantum information theory. After 20 odd years, they've run through most ideas, and we certainly think we can offer them something to think about. The most urgent question for quantum information theory is, of course, to do with quantum computing. Josephson junctions, what are they thinking? The biggest question for quantum information theory is how quantum is life. And it's life that joins us in this. Life does, of course, have natural beginning at the quantum scale, and today presents a unique, strange, and information-rich state of such extraordinary complexity that it's readily expected that living systems will be found to be quantum computing. Quoting from Professor Vedrell's own book, Decoding Reality, perhaps quantum computation takes place in every single living cell. Equally, the fact that there exists a real possibility that quantum computation has been implemented by living systems is a very exciting and growing area of research. And let's hope it is. This can contribute. The widely shared intuition, if well-founded, surely means that quantum information theory should find inspiration in the quantum mechanical analysis of living systems, and possibly therein answers to both questions. Finding a qubit would be a start, wouldn't it? So the CIBSOC humbly presents a gedanken experiment looking for life's qubit. First of all, the experimental system we'll be using will be the human genome. Although it is, well, possibly the most complex of genomes and underpins the possibly most complex of evolved systems, it is the world's best known genome and the data is widely available, so anyone can take their gedanking further if they wish. The illustration on the left is from Alberts et al. 1984, I think, first volume of the molecular biology of the cell, if you're interested. The system components you need to know about are luckily very few. You need to know about DNA and, well, sort of know about atoms. Only 26 atom species are modeled as being involved. One of those, of course, being carbon. Hydrogen, oxygen are the companions, are found in Trinity, and that leaves 23 atom species needing ordination and control in the quantum mechanical environment that is a biological cell. For those who don't have the ready biology, as it were, I've put up the illustration in the middle of DNA base pairing. You can see here where we've got the AT base pairs, which use 2 hydrogen bonds, and GC base pairs, and the sugar phosphate backbone. Over on the left, we have a cartoon just making clear the simplicity of the structure. The telomere is the arrowed head, as it were. These are both adaptations of Richard Feynman's little energy widgets, which one day we'll actually be able to do officially and make sense of it. And on the right-hand side is one of the good old wire models with a, well, I don't know what draped over it, a stocking, I guess, to make a probability envelope for the orbitals. The system dynamics, we only have 2 limit states, which is very lovely. The working state below left here, and the division state below right. The asters are centrioles, they are microtubule structures, they are vital to DNA replicating itself. Without the centrioles, the DNA, duplicated DNA, wouldn't separate. We've got the asters, as I say, we've got the nucleolus here, which appears within the nucleus in the working cell. And we have the metaphase plate, which is signature to the division state. In the cell cycle, I've placed these 2 limit states within the common cell cycle. G1 or G phases are growth, S phase synthesis when the DNA is being copied and replicated, and M phase for mitosis. And you can see where the limit states exist within that cycle. For those, again, without biology, I put the cycle within a cycle of cells just to make sure that you realize the biologists aren't fibbing, and it really does go on. And hopefully, yes, that will make sense to you. You can see we have this original normal resting cell Here, the centrioles have doubled, and that means the DNA is already doubled, and this can begin separation with the asters pulling and making the mitotic spindle before the cell divides. The next thing we need to look at is, well, making a general case for the human genome as an emergent product of bounded wave energy, which is actually very easily done when working at gross scale of gross morphology. But then the genome works at the scale of gross morphology, so why shouldn't it? The staining on this marks 80 rich DNA sequences indicating essential metabolism, which is 45% of activity for the sake of this model. The unstained regions then signifying evolved metabolism, which is 55% of activity. This particular layout from Albert's is, it's weird. I don't know why they ever set it up that way, but we can quickly improve on it by laying it out as double structures. And you can see already that an harmonic arrangement begins to emerge. I mean, you just have to think of organ pipes. Standing waves are what are driving part of this. But it is also immediately clear that this is not the fundamental set, as is indicated by the 5 chromosomes with highly condensed arms, and which, with normal partner arms, these ones here marked with red triangles, look like cricket bats. These 5 are denoted as driver arms and will inspect their working within the limit state that is the working cell. But most immediately significant to me is the indication that our 3.65 billion year old system has grown at the effective rate of 1 base pair per year and evolved into a suprasystem of 5 subsystems operating in parallel. I just have to have a drink, excuse me. Gin is good. Harmonic echoes. Here we see the single arm set laid out, and the improved harmonic is obviously instantly evident, and finds echo in the 5 driver arms when they're laid out in similar fashion. John Dryden knew a lot about harmony, maybe not a lot about other things, but he strikes the right note there, because it does close full in man, it being his genome. If we divide down that set of single arms into 5 subsets, you just start with the longest arm on the left and then overlay them 5 by 5. And each subset should give rise to a solidly stained composite, effectively ensuring the whole set has enough essential metabolism to complete its working. The red line has been placed to show how the 5 subsets coordinate to realize an orderly staggering of each subset's working in evolutionary mode, which are the clear windows. And these periods are recorded in the unstained stretches of the material. They are literally evolutionary windows, as will be seen. I think that illustrates resonance and coherence. So altogether, we do seem to have a QM genome. It's looking like it's QM, certainly. And that would mean that this then has to be a qubit, does it? Is our question. The gedanken experiment allows that life is QM from the bottom up, which means that this illustration of an unusual form of DNA, the G quadruplex, might then be seen as a magnetic ion trap, in other words, a biological qubit. Here it's formed by the interaction of the ends, and you can see these 2 helices are meeting by the ends of 2 chromosomes, and they form a trap. And many hold it simply to be a cap to prevent DNA loss during cell division, which would, well, shorten our chromosomes and supposedly deleteriously, but I think we'll see that's a bit of a fib. Here we have the G4, the G quadruplex. This is the structure we were talking about, and on the left you can see that there is this long extended sequence that goes over the helix partner, and then next to it we have the picture of where it's folded on itself, and you can see the 3 planes that make up the trap. Over on the right hand side we have another angle of approach and vision on it. Left, this is an illustration from Wikipedia, and the atoms are shown by the central green sphere. And it's interesting that it should be on Wikipedia before any quantum trap merchant has picked it up. The computer image used here makes clear how, at the very end of a chromosome, a single strand of DNA extends far beyond its partner, carrying anything up to 3,000 G4 sequence repeats. The strand can then, of course, fold back on itself and form a string of qubits. So in essence, strings of qubits constitute the ends of all our chromosomes. Is it the perfect qubit? Well, you have to think so. This intimate association of trapped atom and DNA surely begs the question as to whatever informational relay, even if not what I'm saying, but there must be an informational relay of some sort made possible by this arrangement. And it certainly naturally prompts the idea of a magnetic ion trap. Aaron Klug pointing out, of course, they readily form in vitro and nature will find a way of using them in vivo, which, well, they certainly do. Let's turn for a moment to the biological perspective, because, of course, the quantum mechanicization of biology is being fought against very hardly by them. But the biologists have been working with the structure since the 60s. It was brought to notice again in the 80s. G4 research is of worldwide interest to the biological community, primarily because of the putative role of telomeres in disease, cancer, and simple aging. Interest, of course, surged again in the 2000s after the, human genome mapping project, and after further analysis revealed the prevalence of potential qubits, sequences, within gene promoters. These are upstream gene switches, effectively, in human, mouse, chimpanzee, and rat genes, so all through the vertebrates, certainly. The finding of potential qubits within gene promoters naturally encourages biological interest, with a variety of lines of attack being explored for the targeting of G4 function. To which I have to say, yikes, because the biologists might not know what they're doing if they actually stick on that. In the biological perspective, continuing, Professor Stephen Neidl at UCL and Sir Shankar Balasubramanian at Cambridge are cataloguing the variety of trap forms, some are illustrated below, as well as the atoms and molecules found therein. Today, most often, monovalent cations, sodium and potassium. You can see these chair and basket, hairpin, and tetrameric forms, but do note they are intermolecular in an intramolecular. Intramolecular informs the chromosome, intermolecular can inform multiple chromosomes. It's a networking issue. Further research has, of course, established the presence of a total of 300,000 canonical G4 sequence repeats distributed not only to build the telomeres of our chromosomes, but then also embedded within the working genome. In 2009, Elizabeth Blackburn and her team won the Nobel Prize, indeed, for discovering all about telomerase, telomeres, and how they protect the chromosomes. And indeed, I think it's from the Nobel site that I took this, they indeed suggest they're protective caps. And the findings are the same, aging, cancer. The thing that surprised me about this was prize-winning research confirmed that an enzyme, telomerase, which is simply a protein, carries an RNA sequence that templates DNA, which ought to cause a discussion about which world is running it, RNA or DNA. But most specifically, it templates the G4 sequence, TTA-G, thereby indicating that the G4 quadruplex is the primary qubit for the eukaryote system. beginnings or ends is interesting because our ends are our beginnings. So whilst the function of telomerase is widely held to be that of maintaining telomeres, it is effectively building chromosomes that are pure G4 strings. The occurrence of G4 units beyond the telomere and their distribution across the working genome surely bespeaks a far more profound function underlying a 400 million years conservation of a sequence that predates the radiation of the vertebrates. Taxonomy provides that 400 million year timeline, but given that telomerase builds the G4, we can extend this insofar as the necessity of chromosome end protection can only have originated in the prokaryote to eukaryote transition of circular to linear DNA, an event estimated at 1.5 to 2 billion years ago. Accepting the G4 as a 2 billion year old ion trap is reasonable when modeling the cell as a quantum system that must constantly monitor its QM environment. And that's to say, the variety of atomic species presenting is being the least of the problem in a quantum mechanical cell. Before we move to look at the working state, this is just to remind you, on the left we have our cartoon chromosome, and on the right, the clear difference there is with the 5 driver arms, 13, 14, 15, 21, and 22. Guess what my numbers are in the lotto? And you can see the nucleolus, which is where these short driver arms embed themselves. The highly compressed sequence is ribosomal RNA coding sequences. There are a variety of them, but they're in high repeat numbers and they make a very nice periodic crystal, a nice long stretch of clear crystalline run. In the working state, the 5 driver arms co-aggregate and form the nucleolus. Their partner arms then extend, and we have the nucleus, and the partner arms extend across the nuclear lumen and anchor into proteins beneath the nuclear membrane. The emergence of the nucleolus offers an invariant geometry that is signature to the working state and offers trap sampling of the presenting ionic array in nuclear throughput, i.e. the quantum mechanical environment that the nucleolus sees. The nucleolar G4 input from here, that is, then naturally combines with the telomeric input from the other end, where you can see again a telomere, and they combine within the chromosome. The rest of the chromosome set members, as with this one pictured down here, just sort of lollygag around and self-organize to occupy the remaining nuclear volume without the nucleolus. So they just self-organize there. There's a thought that needs to be had here, which is about cool running, by which I mean that we're... Sorry, I need to have a drink. Edit that, please, somebody. With respect to DNA, simple QM logic tells us that a whole bunch of atoms or harmonic oscillators only constitute a molecule by the continuous exchange of energy between charge centers. That is, the atoms making up the molecule. Whatever sort of current science might choose to nominate, it is the field this current generates in the trap that holds the atom, and in this much, isolated from all other atoms and molecules, can be regarded as effectively operating at 0 Kelvin. The proposal gained support in Paul Davies' 2008 recalling enzyme experiments, which similarly then suggested a previously unbelievable working temperature of 0.016 Kelvin. Within the nucleolus, the qubit strings of the driver arms sample the emergent ion population simply by trapping. That each G4 string may be formed by one or more of the driver chromosomes serves as a means of information distribution and coordination across the 5 subsets. In other words, we have 5 trap lines sampling the ion population in the nucleolar well. There is no need for anyone or anything to rush about catching atoms in flight and then having to separate fast and slow. Here, the system delivers a free atom, and the G4 subjects it to magnetic ion trapping and maybe even proton streaming to extract quanta, which constitute information as far as the biological system is concerned. Atoms don't like being trapped. They kick off, and that can result in a release of energy, the escape of a quantum, which event can resolve in any one of three ways. It can rejoin the atom, continue in parallel, neither of interest to us. But if the quantum gets out of the trapped cavity and is taken up by the DNA, it's an atom not already in the DNA and can only do so as a phonon, one must suspect. Phononic energy building within the chromosome will then naturally give rise to a standing wave pattern that is overlaid or added to the sequence information encoded by DNA. Phononic uptake by DNA allows additional information to direct subsequent events, insofar as the phononic patterning of DNA, where that is then transcribed as mRNA and in turn makes it to translation into protein, will then produce a protein that has a natural or inherent affinity for the atom species that gave it rise. Thus it is, for example, that the protein calmodulin loves calcium. This would explain how these massive atomic systems can make such rapid and appropriate evolutionary response. The proven flexibility of the G4 has, through evolutionary time, perhaps at least 2 billion years, enabled the trap processing of all the atoms that today constitute our being. Begun in chaos, sodium and chlorine were our original electrolyte. The echo of the sea we evolved from, but potassium offers a greater electrode potential than phosphorus. No, not than phosphorus or nitrogen. It's the first outside of that set. In this light, the trapping of already known atomic species may then serve as a means to refresh DNA and cellular memory. Doubling the information content, one of the differences between natural and man-made computing needs mention. Whilst the physicists trap an atom in order to write to it, as is required for quantum computing, the trapped atoms here are effectively speaking to the DNA via phononic patterning. Contribution can also be made by the intramolecular G4 distributed within the genome. These G4 units interrupt the sugar phosphate spine of DNA, and we might perhaps model them as QM analog of the nodes of Ranvier that break our own nerve insulation and are often proposed as accelerating signal transduction. Certainly, whatever happens in physics happens several thousand times faster than anything in biology. Given that nuclear geometry, when it's in the nucleolar geometry, might allow ion ingress to the DNA interior, we might look on a chromosome as an organismal form, eating ions, a sort of living solenoid. Filling the channels that surround the DNA proton core, ions would then be free to self-distribute against their standing waves. So you could have not only the standing wave, but also ions themselves patterning what's happening. When we go to the normal, the Watson-Crick type DNA helix, best known are the hydrogen bonds that bind the complementary base pairs, as we saw before, AT2 and DC3. And in their bonding, they span the lumen of the DNA helix, effectively building a proton core. And in type B DNA, the form that is actively read, the bases are perfectly angled at 90 degrees to the core to create a proton or ion stream, a ray. That would be the origin of the proton streaming mentioned earlier. So when we look at the limit states, informationally it is proposed that the working state naturally presents a biological maximum, whilst for the QI side of things, it presents a phase building towards the division state, when we expect a QI maximum. Sorry, did I say that? Edit, thank you. In the division state, the condensation and relocation of the duplicated chromosomes to the metaphase plate offers coordination across the whole genome. Plate kinetics adjust to accommodate the vibrational and acoustic energies locked within each chromosome before the set reaches the biologically quiescent two-dimensional state of the metaphase plate. It is during this period of kinetic settlement that a cascade of ions and energies, electric and acoustic, are instant upon the metaphase plate. And as Davies points out, such chaos can act to bring decohering systems back into phase. And one has to wonder, is this then how the system achieves full informational record and closure in this sort of chaotic end of division? We have to wonder at biology and computing as a happy convergence. Schrödinger certainly saw life as offering new physics. It's certainly possible that new physical ingredients going beyond those of 20th century physics might be needed for a full understanding of the physical underpinnings of life. And certainly insofar as even the physicists hold quantum mechanics to be incomplete, that seems quite reasonable. Davies again confirms the uncertainty must be allowed to reign during a period of convergence. In complex states of matter, simple KT reasoning can be misleading, as we have seen, 0.016 Kelvin. The very existence of high-temperature superconductors demonstrates that collective quantum phenomena can take place with a relatively small amount of sophistication and without the necessity of extreme cold. And that's Professor Roger Penrose, and we all know he's no slouch. 300,000 trapped, coordinated, and entrained potassium atoms would probably agree. If we look at the Qubit summary, any and all atoms can be found. I mean, they actually find NH2 has been found, the molecule. So it doesn't matter how big an atom you want to get in there, it'll be fine. In terms of qubit numbers, we know that on the intramolecular, we've got 300,000 distributed across the genome. In the intermolecular, which are the telomeric ones, we've got 23,000. That number comes out because rather than 3,000 repeats at the end, I've worked with 2,000 repeats. Divide that by 4 gives you 500 qubits. And you've got those at each end, at each telomere, so that's 1,000 qubits per chromosome. And with 23,000 genes, you've got a one-to-one mapping between the G4 telomere and the gene. Qubit working intramolecular informs the chromosome arm. The intermoleculars enable networking amongst the 5 parallel running sets. The maths of all that ends up at the total number of qubits in the human body being 9 times 10 to the 18. So IBM and Google aren't looking that hot at the moment. There are also 400,000 non-canonical G4s also found within the genome, and these are mostly corrupted at different base, at different bonding, and different type of net. Taking that corruption as bespeaking disuse, and that these are effectively a paleontologic record, we might be able to realize a means of determining the history of chromosomal assembly. Hopefully, this presentation gives a start to understanding how life might be quantum computing, but we are still urgently need to understand the physical underpinnings of life at its simplest, i.e. just a QM model of the cell. Microsoft promised one in 2015. I don't think we've heard hide nor hair, have we? In truth, we can no longer be sure as to what biology actually is. If you model a cell as 100% QM, which of course it is, that means the biological output, which constitutes only 1.8% of working, might then be seen as noise in an otherwise perfectly operating system. This is nicely poetic, given that the atoms talk to the DNA when trapped. Noise, scream, let me out. As has been shown, whilst the physicists extract an unnaturally formed metal, build motors, develop plastics, and refrigerate a 2 cubic meter box in order to catch an atom, this is something living systems have evolved to do by the gentle deployment of a string of atoms. Such is the power and subtlety of freely self-organizing and evolving quantum systems left to their own devices for 3.65 billion years. And that's why you feel so tired. And that is it, the end. Thank you very much.
Thank you is not even adequate. Sally, I know you've been working hard on this, and every time I hear it, I learn something different.
Okay.
It is
all these people.
They all popped up, right? They're all here and listening
Yes.
intently. And just to say, it is a remarkable challenge for us to think about what cybernetics can offer. And I'm struck by so many things, but we can talk about that as we go along. But the implications across different systems of the fundamentals of what you're talking about is what I find quite fascinating, is interpreting it not only in the quantum sense and the biological and physical sense, but in the organizational and personal sense. So there's a lot of challenges there. You know, I picked up some things.
Peter, we can hardly hear you.
Okay, I'm sorry. Thank you. I'm sorry about that. Okay, well, I'll shut up then. That's good excuse. And let's go to people who've raised their hands, which is great. Okay, Martina, go on.
Goodness, I don't know even where to begin to thank. Sally, I'm a physicist. I know both Paul Davies and Roger, and it's finally somebody that knows how ignorant physicists are. Please, this is so nice that you expressed that our, well, in my understanding, our knowledge of physics is very incomplete. And so is our possible knowledge of hoping to explain biology coming from physics. So that's really good. I really like that because I completely agree. The question that I discussed with Paul Davies about a year ago. He was rather interested, as I have been, on the membrane-based view of the cell. Does this, as opposed to the nucleus-based, does your talk have a relation within this dichotomy, as I understand it? I'm a theoretical physicist. Please forgive me.
No, that's fine. That's fine. I know what you're saying. Actually, we've rather, the society has structured it rather well in the sense that this presentation, the Qubit, is effectively to deal with the nucleus and the nucleolus and the habits of the cell. But the extension of it would be then to extend to the cell, because that obviously needs reinterpretation in physical terms. And we have things like, the plasma membrane is effectively Russian doll packed buckyballs. So we have all the properties of buckyballs in quasi-state and things like that to look at. And it does offer a huge thing. You may well actually have, it's a plasma membrane, but there may actually be a plasma in it. And that's the point, really. That would change things hugely. They're very fixed in biology. They do like things to all be stuck together and to stay there, not move about. But Dell electrophoresis will have its way. But they still don't even interpret their own subject in those terms. The only means where a protein can place itself is by that environment. And they still don't even model that, do they, in biology? But no, you would know, you're a physicist. But they don't, trust me. They're way behind.
Yeah. I completely agree. So please, all the energy to continue to work on these and to show the world how we physicists or our way of thinking is not sufficient. We have to.
No, it'd be lovely. It'd be lovely.
Thank you.
Any other hands?
No other questions? Excellent. I'll be off then.
Well, I mean, the cyberneticist in me, if you can hear me, is just fascinated in the parallels. Maybe I can just take the fact that I'm here to just, the rose window is just one emblem or symbol or icon that you've put forward, and how
The
that
type B
echoes.
DNA, yes.
Yes, I mean, the fact that you mentioned the rose window, and the rose window is a universal symbol in many churches, in many ways representing life, is fascinating to me. That architects and artists, for example, create a rose window, and you're showing us a rose window in
nature.
In nature.
Yeah. Yes.
And so, one of the things about cybernetics that's always I've loved is the ability to move across systems with the same language.
Yes,
and so that
absolutely.
is what strikes me about your work at the fundamental level. I mean, not only just reminding us to think in 3D, but you said some interesting things like chaos can bring decoherent systems into phase, and you think about how chaotic systems can self-organize themselves, for example.
And that was Paul Davies, and he's pretty good at his stuff, being a physicist.
and self-organizing as well. I mean, you're talking about that, these things that people talk about in organizational life, for example, are just fascinating to hear you saying them about it in quantum terms within a living process. That's what fascinates me. So,
excellent.
Have you got any comments on that then, Sally? I mean, we've talked about it a bit, but how what you're talking about gets to some fundamental issues around life itself.
Well, one of the things I like about it is the compelling nature of having an atom in the trap and then generating proteins that are a direct response. In one way, that refers to information. There's lots of energy about this. The thing about entropy, information, and evolution, and there's lots of information about, but it doesn't all get used. So if you get a direct and targeted response as the result of an informational relay, then that information has been fully appreciated for what it is and the response sent instantly back. And that's it means it takes out the randomness from the whole of the Darwinian evolution story. The system is being informed. It knows what's out there and what to look out for and how to deal with it. Unless it's a tiger, by the time it hits the body, they'll be ready for it. But it's it's just it does take away all the randomness. It gives a purpose also to proteins who don't know what they're meant to do. But then suddenly, they're filled with calcium energy, and there's calcium, and they love it, and it's wonderful. So it's worth looking at, because I don't like the randomness in Darwinian theory. I don't think nature has that much time or patience in that way. It has plentifulness, and if you can overcome the whole problem by numbers, sheer numbers, then you're halfway there, because that's the game that nature plays. Numbers always win.
The other thing that, if I can just indulge myself a moment here, is you used the metaphor of organ pipes and the number 5.
Oh, yes, the organ pipes, the standing waves. Well, organ pipes, yes. And literally, they make our organs.
Well, exactly. When you look at word usage, exactly. So there's a vibratory, harmonic, musical quality to our lives in that sense.
Absolutely. Well, if we're all built in string theory, and I don't know if any of the physicists are stringers, as it were, but strings and natural harmonics and music and a dance to the music of time, as is life. So, absolutely fine.
James, let's go to you. You've got your hand up. Please ask Margaret Sally a question.
Thank you. That was really an extensive presentation, Sally. Very intense. I am basically a biologist, but who has spent time with Hameroff and Penrose and Josephson at several conferences, and I've got some experience in, and not exactly in full quantum theory, but enough to understand a lot of it. And I have some concerns in things I didn't quite hear in your presentation. One of them is there's so much dominance with the physics and the quantum mechanics. I did not recognize or sense any cybernetic feedback coming from the phenotype realm back into the genotype. That seems to be a missing part of it. You may have it in there on or in other areas, but it wasn't in the presentation today. That mapping is strange. And the conversion, to put a lot of prioritization on quantum mechanics as being biological really doesn't do proper, because there's so many non-quantum transforms and influences that are involved, that the translation between and the interconnectedness between the two domains of performance is really something that really needs to be discussed, and I didn't quite hear that in the material that you covered today.
I
Just
agree.
a quick example that of simplistic. This is a toy model that I've been using for decades, but it might resonate with you a little bit. That the quantum shells of electrons, say for carbon particularly, because that's our dominant atom, where there are open and shells that can be filled or emptied. And I liken that to non-volitional performance, non-intentional performance, whereas it's basically we breathe, we inhale our version of atoms and energies. Essentially oxygen, give off carbon dioxide, whatever. But that process of taking in and giving off, basically, if you track that down into the physics, that occurs in atoms. Atoms don't breathe, but basically the valence shells of electrons are the lungs of atoms. They're not physical in the sense, they're relational in their performance. But unless atoms, if atoms don't breathe with their performance, we don't breathe, living systems don't breathe at our level of organization. So there's a one transition point where one format converts into the other and they interact quite naturally. So I've got some other things, but those are the three main categories of ideas, of aspects that I'm concerned about. And if you have some comments, I will just close my mic.
I don't know that I do. I'm still having to fight hard to get a quantum mechanical model of the cell, let alone consider the larger case, as it were, before I transition to looking at other systems. But it's, I mean, I work on the basis of 5 internested systems: philosophy, logic, mathematics, physics, biology. And the internesting of those is interesting enough. But I wouldn't know how to answer a lot of your questions because we haven't got that far yet. It's still in development. So I'm sorry.
No, fair enough. I mean, you have what you have. You don't have what you don't have. I'm in the same
I have
summer,
what I have.
so that's fine. Thank you.
Thank you,
You're
James.
welcome.
David, you're muted, please.
Click the right unmute button. Yeah. As usual, and more, thanks enormously, Sully. There's a mass of that I don't understand, haven't digested, need to go over at least half a dozen times, but that's not going to stop me asking questions. There's kind of three things, any of which you might care to riff on. One is, how do we make a load of money out of this, or stop other people making a load of money out of it, which has some possibly sort of traumatic connotations. What are the developmental properties? I mean, part of what you've said implies that really we're infinitely, well, we're a long way ahead of Claude in some respects, potentially, arguably. If we're quantum, if we're quantum information processes, why are we so damn stupid so much of the time? And my other thought is around, well, information theory, quantum theory, entropy, the equations seem to be a bit similar because they're all about probabilities, arguably infinite numbers of probabilities, out of which, or potentialities, out of which things emerge. And there may be situations when you can go back to the infinite potentiality for your calculation rather than just, relate on the sort of everyday thinginess which we've been brought up to think in terms of it. I mean, would you care to address that or go off on a completely different sort of riff?
I think I'd go back to the money. I don't know. I mean, yeah, how do we stop anyone doing I mean, there are ethical issues, obviously, about the development, as it were, as there are with LLMs. But I mean, on this level, what's frightening me is the amount the Chinese are investing in their disciplines. I mean, the Chinese are investing in chemistry sufficient to outpace the UK, the US, and Europe all combined. And that's an astonishing investment. And of course, they have no, what can I say, moral bar on building or attempting to build autonomous organic systems. And that's the sort of thing I do worry about. So it's a case, I guess, of control and keeping your eyes open and understanding what they can do with it if they take it away and prove any part of it. Because if one part's okay, then probably a lot of it is in its fractal way. The thing about atoms, actually, sorry, going back to James Rose, you were talking about atoms and their electrons and shells. It does have to be, the whole transition needs to be to fractal geometry, of course, because that's the mathematics you should be using if you're doing anything with nature. You can't use pure mathematics, to my mind, unless you're doing things with man's interference. If there's no interference by man, you should be using fractal geometry. And it's astonishing to me that the academic community haven't taken it up more readily. Santa Fe was founded in 1994, and they've done nothing that I know of note. I'd be interested to hear if they've done anything profound, which they should have done by now. And so they've had 2 iterations of the idea. I work in 15-year steps for the history of ideas, which chimes nicely with the professor's career. 15 years developing yourself, 15 years at height, 15 years mad emeritus. And after 30 years, yeah, I don't see what Santa Fe are doing. So I think they've missed the trick in some way or other. They don't seem to realize the potential of the maths.
I had a head with Stu Kaufman several years ago, and as much as he made claims to photocatalysis as being a key solution, he really failed to accomplish anything either. He got a lot of funding from Citibank and other major investors, but that was afloat and he never really produced. And most of Santa Fe have some wonderful people there, some brilliant minds.
Yeah, I'm sure there are, but it would be nice to see, for instance, a tulip, a modeling of a tulip growing. It would be simple enough. They've got the L system, which is a cheat, but if they could manage to break through and realize how they're meant to be using the geometry better, we could have a model of, a tulip growing from a bulb, and that would be lovely. And a good computer should be able to do that. Maybe it takes a quantum computer to do that, though. I don't know.
I've got a sense that there's some very subtle things that we have in our mathematics, which is foundationally incomplete, and I've been exploring there too. One of the things being that there's a break between scalar and tensor mathematics, and which I think is doing a conceptual block. It's a very subtle one, but it's something that I've been exploring too. But it's the correlations between the two bodies. One of the things is Dean Falk, who's an anthropologist, did studies of the Australopithecine development of 300,000 years ago, down in Africa. Did some interesting work back in the 90s, and I found that I did an analysis of her work, and I had some contact with her, and we sort of agreed that there are times when you can make a single change in a single genome, such as a stop codon, that has an effect on the physiology of in a developing fetus or of an organism. And so that changes the phenotypic patterns that can have eventual result in behavioral performances. And so there are changes which are not exactly, the old model of genetic mapping of genotype to phenotype.
No.
It just that's not the only.
And don't forget the homeobox genes as well, which Nuslein Volhard and people were doing in the early 90s, where you've got manager genes which aren't doing any protein creation, but they are doing management of other genes, which is the butterfly effect we wanted. So although I was told by my supervisor it wasn't possible, it's actually there in Utrasoft and nicely done. Yeah.
Maybe we can take the
Sorry.
No, yeah, I was going to hand it to Richard.
No,
I was going to say maybe we could take the tulip modeling idea to the bar, and it's one of those things we might come up with. Richard, you've got your hand up. Richard Helen, please.
Yeah, hi. Thanks. My first time here. It's very interesting. One of the advantages of being kind of dilettante in this sort of ecosphere or ecosystem of interesting ideas is you can jump across from one thing to another. And I'm wondering, and picking up what Peter was saying about sort of spotting similarities. I'm wondering how the connections might be made in your mind, Sally, between your work. You mentioned Penrose, and somebody mentioned Hammeroff with their microtubular world. And quantum biology with entangled processes. And the other one, which is a little bit weird, and I don't know if any people know about this, is the work that Michael Levin and colleagues, he's at Tufts doing some interesting stuff in developmental biology, including making, I think, what he calls them as cytobots. In other words, making small creatures out of human skin cells, and they have all sorts of interest.
Xenobots.
Xenobots, I beg your pardon, xenobots. You're absolutely right. Yeah. What do I think? Anyway, but so what I'm getting is a sense of, wouldn't it be interesting to somehow get these, let's call them non-mainstream ways of looking at stuff and seeing how they resonate with each other, to say, hang on, what you just said is very similar to what I've been saying, and I was using different words. Maybe we ought to, get together and have a chat about it. So that's just my observation, and it's absolutely fascinating, and I'm really keen to find out more.
Well, are you a member of the society, Richard? Because I always think that's what cybernetics is about.
Thank you.
Yeah, good. Any other questions or thoughts here? We can always.
Good. Excellent.
Excellent. I will say.
Einstein said one good argument would be enough. So if nobody's arguing, that's fine.
I wanted to defend Stu. I mean, Stu Kaufman. I think he is no longer associated with Santa Fe, as I believe it. If I believe it, or if I understand it correctly or not. He does have a new book, which I was just reminded that I've not yet got back on. Thank you for bringing that up. So he's my dear friend, and so if I'm going to criticize him, I want to criticize in a good way. That although he thinks as a systems biologist, he is still secretly a closet mathematician, so to speak, as our physicists. So although we also try not to be mathematicians, I think we are closet mathematicians. And so, but I believe, and I did think that Mike Levin's work was rather mainstream. But as a physicist, I don't really have a good grasp on what is mainstream in biology or not. So if you will care to give us a view or a perspective in biology, what is mainstream in that sense, because we don't, physicists, I mean, I'm a cosmologist, so we don't talk to you guys much.
No, you don't. But Paul Davies does, and he's very good at
Yeah.
talking to the biologists. And it's difficult to know what to say. I mean, you do have to work alongside each other. There was briefly in this country a research council set up to do the interface of mathematics, biology, and physics. And I sent them a poster, which was chaos fractals and the quantum mechanical organization of the human genome. And they wrote back and said it didn't fit with their remit. Well, I just don't understand. I mean, I can't help them there. You can't explain. So what do you do? It's just lovely. I mean, the thing that is good about the Cybernetic Society is there is no axe to grind in terms of systems. So they don't say anything is wrong, which is how I got accepted, how I got to be a member, and how I got made a fellow, because that's fine. It doesn't matter what you are, as it were, as long as you're thinking in terms of systems and manifold, embedded, complex systems at that. And cybernetics, of course, as Ashby said, is the science of complex systems. And so that's no answer to anything, but it's still nice to say.
It's a beautiful answer. I think it's a beautiful answer. Yeah, no, thank you. Richard, very.
Thanks, Peter. I'm just going to try and play back what I've heard today, and I've kind of put a couple of questions in the chat earlier. The first one is, and I think we're starting to excavate, and that's around the gaps in the cybernetic knowledge. We can talk about physics, we can talk about chemistry, we can talk about biology, we can talk about quantum, but is there something cybernetically relevant and different at the lowest level of recursion that we're missing? It's just hidden in plain sight at the moment. So for me, that's just a really interesting topic that's almost come out of the questions and answers today. And the second one is perhaps for later in the bar, but what do we do now? This clearly has been a very impactful session, and it's continuing to be. But how do we take this forward? We're identifying gaps, we're identifying opportunities, we're developing propositions. Okay, so what happens next, and what's the role of the society in taking that forward? And I don't have answers to those questions, but I'd be most grateful for any thoughts that people have. Thank you.
Well, for myself, I would have to say, I mean, as I did refer to earlier, we've nested it nicely where we've got the nucleus and the nucleolus. They plop into the cell. So once we've got a better understanding, as it were, of the dynamics of the cell biologically, the answer to your question about the levels and what refers in cybernetic terms will become more apparent, but we still haven't got to the bottom of the cell anyway, so that needs looking at. And when we do the cell, as we discussed, I'd like to take it to the first suggestion that came through after Schrödinger gave it to the physics guys, which was superconduction. And if we have a superconducting cell, that changes everything and makes it much more manageable. But in terms of the modeling, that's the only way I can think of things at the moment is in terms of computer modeling. And there is as I have pointed out to others, there is the Zindo mapping, which gives you effectively what I imagine to be what DNA would look like as a type 2 superconductor. And that is sort of the technical side of things, I guess, that we need people to help with, really. Technical, I mean, equipment, we don't have any equipment, we don't have any, computers or anything. I mean, so it's somebody who's got computing power, maths power, and knowledge of physics, and is happy to give us their time for now, and that's a start, but it transcends that eventually, of course.
Wonderful, thank you. I think Marina's about to offer all of those facilities and support, hopefully.
Okay, this is when I step to my other career as a ballerina. So I know no answers. I do have questions. So the next steps, and I'm speaking as a physicist, if you forgive me for that. If I had the time or energy, I do believe that to make a better bridge between physics and biology, I'm rather intrigued by Michael Levin's work, but we are so skeptical in physics that it would be amazing. And I'm speaking of myself, even though I try to be open-minded, I always find biases in my own mind and thinking that bring me back to my prejudices as a physicist, that I want things to look a certain way. And so it would be a very interesting project if Michael Levin's work were to produce a catalog of data that we could then have different communities look at. I've been recently at Berkeley Lab because we cosmologists work there. They were looking for new experiments, and I said, well, we need a physicist or a community of physicists with enough open-mindedness to look at these extraordinary effects that Michael Levin is obtaining that I have no explanation for, but neither do I have the or time that would be needed to make a concerted effort towards that. Thank you so much.
That's okay. That's okay. Schrodinger is with you. He's just put his cat in the back.
Yeah.
So just to let you know, Nirosha Murugan, who worked with Michael Levin, and she has her own quantum biology lab in Canada. Yeah. She's agreed to give us a talk next year, early next year.
Excellent.
So that'll be a wonderful way to get into the cell. And her husband does mitochondria, and I'm hoping he agrees with the energy resistance principle that he's talking about, because that's the psychobiology with the mitochondria. So we're trying to build a whole chain to help us with scales and the connections, the recursion transition places, how those could be working. So hopefully we can get there, right?
Yes,
Beautiful.
absolutely.
Perhaps end the formal session, if I might. Sally, have you got any other thoughts for the recording before we?
No, I don't think so. Don't think so, except for the thing about telomerase that fascinates me. There's often in biology, and that's when you're doing just pure biology, they often talk about which world is it, an RNA world, a DNA world. And I think telomerase is an astonishing example of a single protein that is so important. There is only one telomerase for the whole of the eukaryote kingdom.
And you know what's so amazing, Sally, is that nitric oxide is absolutely essential for building telomerase. And we need UV to make the nitric oxide.
Oh, right, okay.
So it's this beautiful
light.
continuity, this chain of, so from the light, the UV signal through the process to the telomerase to, it's, sorry, getting a little bit poetic there, but
no, it is poetic. It should be poetic. If it's true, it will be poetic.
So I'm interested in that 10 to the 18.
Yeah, the 23,000.
So I'm really interested in the other things. I sent you a little link on the other things that are around about that number.
So you were talking about the Avogadro number, 6.022 10 to the 23.
Yeah.
It's a well-known and loved number, but yeah.
So is there anything else that is 10 to the 18?
10 to the 18?
Did you say 10 to the 18 or 10 to the 23 on your
23. 23.
I thought it said 18. Sorry, beg your pardon.
There was.
Yeah, so the 10 to the 23. And the reason for that is because, somebody has to help me with all the little letters that come after the number. It's 10 to the 26, I think, photons per millisecond per blah, blah, blah that hit the Earth. And they're 10 to the 26 mitochondria in the human body.
Oh, right. Okay. One-to-one mapping.
Yeah. And here you've shown us another one-to-one mapping. So I'm looking at, we need somebody who deals with scales, these kinds of scalar things.
Yes.
Somebody,
Yeah.
please. Jeffrey West, please, or somebody like that, who can help us find these scale relationships. That, I think, is also really important.
Yeah.
And
Might
because
like
likely they're
Tim
going to
Palmer.
Yeah, somebody. Yeah, we should maybe
Tim
ask
Palmer in Oxford. He loves.
Tim Palmer.
I guess if you're with the scales, you'd be with the fractals.
That's
And he told me he was writing a new book. His last book was The Primacy of Doubt. He has been rather quiet, but I think he's writing a new book, and the primacy of doubt was indeed about the idea of a scale and the invariance of scale, and he even has a cosmological model about fractals and attractors.
Yeah. Now you're talking.
It's rather lovely.
Lovely. Okay. Anybody got some philosophy to offer?
I was gonna just respond to Richard Helen, if I may. You said something about a platform to continue. Well, I just wanted to add that this presentation has come about as a result of us hanging out in the bar, which we'll do in a minute, and just discussing and learning more about each other. And then we just formed a self-organizing group afterwards, and that has emerged into what we're witnessing today. And so that is, I think, part of the role of the Cybernetics Society, is to nurture that kind of thing. And so we are developing platforms and ways to do that, and it is intentional. And we are looking at how the Cybernetic Society plays a role in that, and Loughborough University in particular, with Peter Kavalak, who's the president, who I omitted to say sends his apologies, but he's helping his daughter at university move out today. So that's why he couldn't join us, and I needed to get that in here. But so we'll go to the bar and let's hang out and see where we do take this. And again, thank you, Sally, for working on this for months. I know, wonderful challenge to us to step up. Thank you very much indeed. Thank you all for attending and these great questions. And we look forward to seeing you again on one of these Cybernetic live events.
Okay, lovely. Thank you.
Thank you,
Thank you,
Sally.
everyone.
Thank you, Sally.